Welcome class to chapter 21. This is on DNA and biotechnology. This is the beginning of our last module or last unit. In these four chapters, 21, 22, 23, 24, the videos again will be relatively shorter, but you can work through the material through those dynamic study modules and through the homework questions on your own. So this is DNA and biotechnology. We'll be looking a lot about DNA and how DNA is replicated, some mutations, epigenetics, which is really popular right now, regulating gene activity, and genetic engineering along with that. So DNA is deoxyribonucleic acid. It's double-stranded to form a double helix. It resembles a ladder. And it's made up of one sugar, which is deoxyribose in DNA. We'll talk about how it differs from RNA, a phosphate group, and a nitrogenous base. The DNA has four nitrogenous bases: adenine, guanine, thymine, and cytosine, and they're all labeled with their appropriate letter. The rungs of the ladder consist of the pairs of nitrogenous bases that are complementary paired together. Adenine always pairs with thymine, so you'll always see A and T together, and cytosine always pairs with only guanine. And each pair of bases is held together by hydrogen bonds. That's important to note because hydrogen bonds can be easily dislocated, so the DNA can be replicated. And genetic information is encoded in the exact sequence of these bases. So this is a great video on looking at the form of DNA. And here it just shows how DNA is a double helix wound together, and then each colored represents a different base paired, and you can see how complementary base pairs go together. And you can see that zoomed in on the right of this picture here. DNA must be exactly copied before both mitotic and meiotic cell divisions can occur. This begins when an enzyme breaks those hydrogen bonds down, the two strands of the double helix unwind, and then the nitrogenous bases are separated. Free nucleotide bases in the nucleus then attach to the exposed complementary bases. DNA polymerases link sugars and phosphates, and as each of the new double-stranded DNA molecule forms, it twists into a double helix. So we call this semi-conservative because in each of the new double-stranded molecules, one original parent strand is saved and is attached to a new daughter strand. Complementary base pairing creates two new DNA molecules that are identical to the parent molecule. And this is how genetic information gets passed down from parent cell to daughter cells. And you can see that here. Here's the parent molecule unwinding and a new daughter strand forming. How does DNA direct cellular activities? Well, the replicated DNA codes for RNA, which will code for proteins. And proteins are what play a very structural or functional role in each cell. A gene is a specific segment of DNA with instructions for producing a particular protein, and it is expressed when the protein it codes for is produced. So RNA synthesis is also involved in gene expression, and RNA differs from DNA in that it's made up with a ribonucleic acid. It's single-stranded, and instead of containing thymine, it contains the base uracil, and it contains the sugar ribose, not deoxyribose. So you should know the differences between RNA and DNA very well. Here's a great table describing those differences. RNA synthesis occurs first with transcription. This is synthesis of messenger RNA, which we label mRNA, using DNA as the template. So mRNA will carry DNA's instructions for synthesizing a protein from the nucleus out into the cytoplasm. These are the steps in transcription, and I'll let you read through these. Again, we're creating a copy of the DNA, but in RNA format. So instead of thymine, we'll see uracil pair with adenine. You'll read through what promoter is, what the RNA polymerase is, and then there's another sequence of bases that will be the stop signal in that gene expression. So this is a look at the DNA, again, parent strand unwinding and how the RNA begins with the RNA polymerase and the RNA forms attaching to that DNA parent strand as the DNA template. And the RNA transcript is released from the DNA. And that can be seen down here as a single strand because we remember RNA is only single-stranded. Modifications to RNA, I'll let you read through these. Introns are intervening sequences that will be removed. And exons are express sequences that are left to direct protein synthesis. This is a great video. I would encourage you to watch this one on gene expression. And this is just shown how the introns are taken out and we are left with exons, and that is the modified mRNA model that will be involved for gene expression. So again, encourage you to look at the differences between mRNA, which carries the DNA information from the nucleus out to the cytoplasm. Then we'll get to tRNA, which binds to a specific amino acid and transports it to be added to a growing polypeptide chain. And the rRNA combines with the protein to form ribosomes, and this is where synthesis occurs. So then we get to translation. We talked about transcription is going from DNA to RNA, and then RNA leaves the nucleus. Translation converts the nucleotide language of an mRNA into the amino acid language of a protein. Then genetic code converts the DNA base sequence into an amino acid sequence. And a codon is a 3-based word in the genetic code. It's made of 3 base sequences that translates into 1 amino acid. And proteins are made up of 20 different amino acids. So for example, the codon UUC, which is a uracil, C would stand for, let me come back to that one, on mRNA specifies the amino acid phenylalanine. So these different 3-worded codes make up the different amino acids, which go on to make up the proteins. There's stop codons, which are universal, UAA, UAG, and UGA. Excuse me, the C is cytosine. I had a little blank there for a second. You can call it my pregnancy brain. And then UAA, this is uracil, adenine, guanine. And again, you don't see thymine because we're dealing with RNA. The stop codons do not code for an amino acid. They just kind of tell the translation step to be finished because the protein is done. This is a great look at how we go from DNA to mRNA to tRNA to a protein. This is how we use this chart to find whatever comes in the first base is on this column. And then we look at the column of the second base, and then we line it up with the column of the third base. And you can see the different combinations of codons that we can get to get the one of 20 different amino acids. And you can see these amino acids listed here. The start codon will always be AUG, and then these are different types of stop codons. Transfer RNA then converts the genetic message carried by mRNA into a particular sequence of amino acids. So it'll recognize both the codon on the mRNA and the amino acid specified by that codon. So tRNA basically brings in the correct codon, and we call the anticodon a 3-base sequence on the exact tRNA that's complementary to the mRNA codon. So your tRNA is just going to bring in this anticodon that will be complementary to the mRNA codon to bind for a protein. It's important to note, though, that the codon itself on the mRNA is what we'll use to translate for the protein. So this is what the amino acid attachment site looks like. The anticodon binds to the codon on the mRNA, and you can see that happening here. And just the anticodon brings along the amino acid that, again, will be complementary to the mRNA. Ribosome is this 2-subunit structure, each composed of an rRNA, ribosomal RNA, and their function is to just bring a tRNA bearing an amino acid close enough for the mRNA to interact. and that permits alignment of an anticodon and codon. So these are the two subunits of ribosomal RNA. You can see where the tRNA will bind and the mRNA groove that will come in and attach to that anticodon of the tRNA. These are the three stages of translation. Initiation is when everything comes together, the subunits, the mRNA and the tRNA. So here's your mRNA coming through the small subunit, the large subunit binds on, and then we get tRNA, which again contains the anticodon, which will line up with the codon of the mRNA. And at the end of each tRNA, we'll have the amino acid that it's bringing in. Elongation just occurs as the enzymes continues to grow, or as the chain of amino acids continues to grow into a protein. And this is just a look starting with number 1 of mRNA coming in. The ribosomal RNA subunit kind of is the manufacturing point, and the tRNA is bringing these amino acids that continue to grow on the polypeptide chain. And we just go through basically this process of tRNA bringing in amino acids, linking together based on the codons from the mRNA. The direction of translation, again, is going this way. Termination occurs when a stop codon moves into the ribosome. And again, this is a great video to watch because I know this is a complicated process, and watching videos on this will help. The stop codon moves into the ribosome, and then release factors cause the release of the newly formed polypeptides. You can see that's located up here. That's your protein of amino acids, and the separation of the subunits and the mRNA. Changes in DNA is called a mutation. So this can be passed to the offspring only when they occur in a gamete. It can be chromosomal mutation, which occurs when whole sections of chromosomes are duplicated or deleted, or a gene, which is a change in the order of a nucleotide in the DNA. These could involve an insertion or a deletion of nucleotides, and it can greatly change the result of the protein. So sometimes a mutation can be a change as a single of one base pair. And you remember from that chart, if we change one base pair, we get a different codon that will form for a different amino acid, which will totally change the resulting protein. So this looks like this is original DNA, but with an incorrect pairing, and how we can have two different possible outcomes based on that incorrect pairing, that one base pair mutation. Epigenetics, regulating gene activity. Only certain genes are active in a certain type of cell, and most genes are turned off in any given cell. This leads to specialization, where active genes produce specific proteins that determine the structure and function of a particular cell. Gene activity is regulated simultaneously at several levels. The chromosomal level is the coiling and uncoiling of DNA. When DNA is tightly coiled, genes are not expressed, and then transcription of genes. Enhancers increase the rate of transcription of certain genes, and steroid hormones can turn on specific genes. Biotechnology is the field in which scientists make controlled use of living cells to perform specific tasks, and genetic engineering is a subset of this biotech field, which involves the manipulation of DNA for human purposes. Some uses are to produce drugs, hormones, improve treatments of human diseases, and increase food production from plants and animals. So if you want good job security, you can go into genetic engineering. Recombinant DNA is DNA made from two or more sources. It's created when a gene of interest, one that produces a useful protein or trait, is put into another piece of DNA. This is a great look at genetic engineering. Steps in creating recombinant DNA: you need a restriction enzyme, plasmids, and a vector. And I'll let you read through those steps here. Continued steps are listed here. And this is a good video, again, about those steps in genetic engineering, which I'll let you guys look through on your own. And you can see them as well here. Again, very interesting recombinant genetic engineering. If you're interested in genetics and you really like when you get if you take a genetic class or microbiology, I would encourage you to pursue that field. Not my specialty, but it is very interesting and it's fascinating. Applications for genetic engineering we talked a little bit about, but there are many, and that's why this field is growing. It's the hot topic. There can be environmental effects where we are looking at oil-eating microbes that can withstand ocean conditions to clean up oil spills. Livestock, cows that are producing more milk, which, no one wants anything genetically modified. We'll get down to agriculture. Crops can be genetically engineered to produce a higher yield. And again, no one likes genetically modified, but when we talk about combating world hunger, there's a lot of people who are for genetically engineered things to help combat the hunger crisis. Pharmaceuticals are gene farming, creating animals that produce a protein with medicinal value. And then raw materials, goats and bacteria, can be genetically engineered to produce spider silk proteins. And we call that farming with a P. So just examples of looking at how they might inject the human gene of interest into a donor cell, implanting the cell into a mother, identify transgenic offspring, and then collecting milk, separating the milk from proteins, and then isolating a useful protein. Gene therapy is putting functional genes into body cells affected by a mutant gene. They use a virus used to deliver a healthy gene. There's potential here to cure some inherited diseases that are listed there. So very interesting things in the medical field happening right now. A genome is the entire set of genes carried by one member of a species, and genomics is the study of entire genomes. The Human Genome Project has sequenced all of the DNA in a representative human cell, and they found that the human genome consists of between 20 to 25,000 genes, and humans are identical in 99.9% of the sequences of their genes, which is totally crazy. So 0.1% is all that we differ by, which gives us all our different characteristics. Epigenome consists of chemicals that bind to specific genes and turn them on or off. And unlike the genome, the epigenome can be variable throughout life as it's impacted by diet and stress. So the epigenome, you can kind of control a little bit based on your diet, stress levels. So a lot of your health is determined by environmental factors, outside factors. A microarray is a tool used to analyze gene activity under different conditions, where thousands of DNA sequences are stamped into a single glass slide. This can be used to identify cells that are active in cancerous cells, but not in healthy cells. You can compare genomes of different species, and that could provide a common evolutionary past. We share 50% of our genes with fruit flies, 90% of our genes with mice. When you think about it, though, that's not a lot yet, because just with each other, we share 99.9%, and we're still incredibly different. So this is a look at that microarray, looking at prostate cancers versus normal tissues and the genes involved with those. So from here, you should be able to do these steps. Thanks for listening, and I hope you guys are doing well.